Good Wednesday morning. A reminder, you have a test on Friday. And a reminder that the test covers lecture material from certain dates. And certainly moving forward, which is also a test next Friday, the tests are covering material that sort of spans the different lecture sections or weeks, however you want to put them. So there's lecture section 6, 7, 8, 9, 10, but things are not, the material on the test don't cover those things specifically. Right? For example, the first lecture on February 24th covered partly microtubule that covered the end of mitosis. Right? So the material covered on the test isn't specifically section 7 or section 8 or whatever. It's whatever I covered on those dates, including whatever I'm covering today. Okay, so the test is basically written, although I have to kind of make sure that I cover material today that's going to be on the test. I think the test is easier than last time. We'll see. I will warn you to read things carefully. I'm not trying to be tricky, but words are my only tool, right? So polymerization, depolymerization, microtubule, microfilament, intracellular, extracellular. Read all the words carefully, right? That's the whole point of this, is I'm using words to make sure you understand the difference between a microfilament and a microtubule. So don't rush through things so much, read things carefully. Some of the questions are dead easy. Some of them are going to require some thought. I do have extra office hours tomorrow from 10 to noon if people want to pop by. Okay, so because of the way things are structured, I'm only going to cover a few slides today that are going to be on the test, so we're going to get out of here probably early. I know that's going to be horrible for you guys, but less material on the test. All right, let's sort of back up a couple slides just to go through this a little bit better. Because I didn't quite finish this with the afternoon lecture, so I might as well go through things again with you guys as well. Right, so cell migration involves cell polarity, right? So there's going to be the front end of the cell, that's where the chemoattractant is binding to some sort of receptor, and things are going to polymerize. Actin is going to polymerize on the front end of the cell. So there's 3 molecules that are activated sort of in sequence that hopefully are pretty straightforward, and those 3 are CDC42, RAC, and Rho. Right, we've discussed those molecules previously in terms of actin polymerization in non-migrating cells. Now this is in a migrating cell. They're still involved in actin polymerization, but they're making very specific structures. So those structures are related to cell migration now. So CDC42 is usually activated first. It's going to be activated by that cell surface receptor that's binding to that chemoattractant. You're going to activate CDC42. It's going to do 2 things. One is activate this gene called PAR6, which is on the next slide, and that's involved in cell polarity. So CDC42 is basically defining the front end of the cell. So CDC42 is going to be in its GTP form, active in what will be the front part of the cell, and that's where you're going to get active actin polymerization, as well as microtubule polymerization, which we'll see on the next slide. Okay, CDC42 is also involved in activating WASP with ARP23, and you're going to get polymerization of actin resulting in filopodia. Okay, so CDC42, you get membrane protrusion, filopodia, as well as polarity of the cell because of this gene called PAR6 that's going to be at the front side of the cell. At almost the same time, you're also activating RAC GTP. RAC does almost the same thing as CDC42 in that it activates ARP23, except RAC works with wave. ARP23 in this case is going to result in lamellipodia formation. So you're polymerizing actin, making lamellipodia, pushing the membrane forward in that direction. Okay, different cells use different combinations of filopodia and lamellipodia. Most of them do it at the same time almost. That's why you have these arrows and sort of signals going both ways. So cells are going to push membrane forward using filopodia or lamellipodia or both, or combination of both. But in general, it's considered CDC42 is first, and then RAC, and you're pushing membrane forward. Once you push the membranes forward, you're going to start making new focal adhesions, and as you start making sort of focal adhesions, you have to then make all the material needed for focal adhesions and stress fibers. So the next gene that's activated is RhoGTP, and RhoGTP is doing two things as well. It's activating formin, so you get actin polymerization, so you get these actin filaments, unbranched filaments being polymerized. They're going to give rise to the stress fibers. But stress fibers are contractile, so another thing that Rho is doing is it's also activating myosin. Okay, so you have active myosin within these stress fibers, because the stress fibers look like little sarcomeres. So you have sarcomere-like structures in the stress fibers that are activated by RhoGTP, and that's usually away from the front of the cell. Make sense? You need the three of them. If you inhibit any one of them, you get reduced cell movement, as I described last time with the scratch wound closure assay. If you make a scratch, cells will try to fill in that scratch. If all three genes are present in the control, you close that scratch, the cells are moving. If you mutate any one of these, you do not get as robust cell movement. So the best cell movement, you need all three of these genes. Okay, as I mentioned, CDC42 is involved in cell polarity, and that's involved in this gene called PAR6. So PAR6 is activated at the front end of the cell. It does lots of things, but the most important one to kind of consider is you need material at this front end of the cell. This front end of the cell is polymerizing lots of actin, making filopodia, lamellipodia, whatever it's making. You need actin monomers, you need things like profilin, you need many new components at this front end of the cell. So that all has to be transported up. And for that to occur, you need microtubules. A lot of translation and other things is occurring in the cytoplasm. You're translating and you're making G-actin, you're making profilin, you're making all these proteins that you need. You need to transport them to the front end of the cell. That's where PAR6 is involved in sort of directing microtubule polymerization so that you can transport material to the front end of the cell. Okay, so CDC42 is at the front side of the cell, it's activated at the front. Then you're activating RAC, then you're activating Rho. And Rho is involved with the stress fibers. Easy? Okay, so that's sort of ending the actin part of it. Next, we're going to describe the last sort of component of the cytoskeleton, which is intermediate filaments. They are the least understood, and they seem to do the least amount. So we'll talk about intermediate filaments, I think, in 7 slides here. Then later on, we're going to switch into cell adhesion. So on Monday, we'll talk about cell adhesion, and there, basically all of the cytoskeletal components come into play in cell adhesion. So even though we're kind of talking about them individually, they do all work together, as we'll see here when we start talking about intermediate filaments. All right, so there's a nice little table here of microfilaments, microtubules, and intermediate filaments, which we're just going to talk about. I've talked to a bunch of you, and I've even mentioned it in class, in terms of studying, try to sort of compartmentalize things. So make lists of things according to some sort of criteria. You know, take a list of any motor protein you can think of. Take a list and write anything associated with an actin. Make a list anything associated with mitosis. Right? I think making lists is important so that you have categories, so when you see questions, if the question is about mitosis, you kind of have a sense of what fits in with mitosis. Right? If the question is on mitosis and I mentioned nexin, then you go, no, nexin is involved in, you know, actins, then you know that's BS. So tables like this is sort of what I mean. So this kind of gives you a little summary, you know, intermediate film, actins for microfilaments, what's associated with an actin microfilament, what's associated with a microtubule, and then what's associated with intermediate filament. So each one of them has certain sort of characteristics with them, and you should not get them confused, right? Actin, using G-actin, it's got certain properties. We didn't talk about all of these properties necessarily, but you should still be able to think about actin and how different it is from microtubules and how now different they are from intermediate filaments. So I'm not going to go through the table per se, other than kind of point out these differences here. So the first one is that intermediate filaments are not globular. That is to say that their subunits are not small. You have G-actin, which is globular. It's basically like this little ball shape that has 4 little domains. Tubulin is made up of alpha-beta dimers, which are sort of globular. They're sort of round structures. Intermediate filaments are made up of subunits that are actually quite large, right? The subunits that intermediate filaments are made of are long proteins. And the basic subunit for intermediate filaments is a tetramer. So what happens is you translate an intermediate filament protein, and the intermediate filament protein has a head domain, this rod-shaped structure, and a tail domain. So after translation, 2 intermediate filament protein subunits come together to form this dimer, and then this dimer comes together with another dimer to form this tetramer. Okay? So this tetramer is organized so that there are 2 head domains and 2 tail domains on one side, and 2 head domains and 2 tail domains on the other side. So this tetramer, which is a rather large structure, is not globular, and it also does not have any polarity. Okay, one side has 2 head domains, 2 tail domains, the other side has 2 head domains, 2 tail domains. So when these tetramer, the subunit, comes together and polymerizes, intermediate filaments have no polarity. There is no plus or minus side to an intermediate filament. Okay? These tetramers, when they come together to polymerize, they don't require ATP or GTP. So there is no ATP or GTP bound to these tetramers. So they polymerize. They don't have polarity. They don't need ATP, GTP. They don't have any known motor proteins. Nothing seems to be transported along intermediate filaments. And they are less dynamic. With actin and tubulin, there's lots of mechanisms in place that you can polymerize and depolymerize them. With intermediate filaments, they do polymerize and depolymerize, but much more slowly. What intermediate filaments are good at, and it's in here somewhere, great tensile strength. Okay? So basically they can act like ropes. You can tie things together and you can hold things in place. So intermediate filaments' main job is usually structural, for tissue integrity, which we'll talk about in a second. Actin and tubulin can do lots of things. They can be structural, they can move things, they have lots of different roles. But for intermediate filaments, their only role seems to be, because of this great tensile strength, They give structure to tissues. All right, so you have intermediate filaments that form a tetramer, and that tetramer polymerizes to make these filaments that give structure to tissues. Those tetramers don't have any polarity. Those tetramers provide strength to tissues. For good or bad of it, though, these tetramers are also tissue-specific. So there are certain intermediate filaments, and if you identify them, you can tell the tissue, right? So often in disease cases, if you see some sort of tissues, like some sort of malignant tissue, if you look at the intermediate filament, you can tell what type of tissue it is because these intermediate filaments are tissue-specific. So there are 5, I think, intermediate filaments I'd like you to memorize. One is keratin. So yes, there's different types of keratins. We're not really going to bother with the different types. Keratins are found in epithelial cells. Right, so your skin, your epithelial cells, you probably know it's keratinized. So keratin is the intermediate filament in epithelial cells. So if you have a cancer cell that's derived from epithelial tumor, it'll have keratin in it, for example. And that's not to say that cells can't necessarily transform themselves, but in general, you can use intermediate filaments to sort of identify the cell type. Okay, so keratin is found in epithelial cells. There's lots of different types of epithelial cells we'll talk about in a second. But a lot of them, they all have keratin in it. Keratin's job is to hold cells together, to form structures. Yes, as the cells die and slough off, you have this keratinized epithelial stuff that is kind of constantly being shed by you. But initially, keratin is an intermediate filament that's used for structure. Okay, so keratin is found in epithelial cells. Another important intermediate filament protein is called desmin. Desmin is an intermediate filament protein found in muscle cells. So inside your sarcomeres, you have lots of structural things, including desmin. Desmin is in your muscle cells to provide strength. So desmin is a muscle cell-specific intermediate filament protein. Another protein is called vimentin. Vimentin is present in mesenchymal cells. What is a mesenchymal cell? For our purposes, it's a migrating cell. It's usually a poorly differentiated mesodermal cell, but we're not going to go there. If you see the word mesenchymal, it's migrating. Okay? So migrating cells are going to have vimentin in them for purposes of this course. So desmin is found in muscle cells, keratin is found in epithelial cells. Epithelial cells, by the way, are not migratory, right? Your epithelial cells form barriers. So if you have keratin, you're probably non-migratory. If you have vimentin, you probably are migratory. Another important intermediate filament protein is something called neurofilaments. As the name suggests, these are found in neurons. Neurons are long structures. You have large neurons all over your body. They are supported by the intermediate filaments in them, including an intermediate filament called neurofilament. There's different types of those, but you don't need to memorize those names. So all of these, so far, so the 4 of them, keratin, desmin, vimentin, and neurofilament, are cell type specific. You can tell the cell based on that intermediate filament. However, there is a class of intermediate filament that's found in all cells, and this is a class of proteins called lamins. Lamins are intermediate filaments that are found inside the nucleus. They're inside the nucleus, supporting the nuclear membrane. So all cells have lamins. Nuclear lamina is sort of one of the names that derives from them. So inside the nucleus, inside the membrane, inside the double membrane of the nucleus are these proteins, and they hold the nucleus shape, so it allows the DNA to be transcribed, etc. Okay, so intermediate filament proteins are cell type specific, and they have great tensile strength, and they're involved in supporting things. For them to do that, though, they do have to interact with other proteins, and they do. Even though they don't have motor proteins, there are a number of proteins that associate with intermediate filaments. So these names are called this nondescript name of intermediate filament associated proteins. Just like you have MAPs, which are microtubule associated proteins, you have intermediate filament associated proteins. Okay, so again, there's no motor proteins, there's no capping proteins, there's no severing proteins. These proteins, these intermediate filament associated proteins, basically hold things in place. An important one, for example, is this protein called plectin. Plectin helps link intermediate filaments to other components of the cytoskeleton. In this example, it's a microtubule, right? So you have microtubules in red, you have intermediate filaments in blue, and in green you have plectin. So plectin is kind of cross-linking things together, holding things within the cell. Okay, so again, you don't have motor proteins, but you do have to link these intermediate filament proteins. If they're going to provide strength and support, they have to hold things, and indeed there's lots of proteins that bind to intermediate filaments and bind them to other things. This also holds true in the nucleus. As I already mentioned, in the nucleus you have lamins. So there's actually three types of lamins. There's lamin A, B, and C, and they are found inside the nucleus, supporting the inner membrane of the nucleus. These lamins have lots of different functions, and they can also support the nucleus membrane because one of them, lamin B, is actually tethered to the nuclear membrane. So lamin B is linked via something called crenellation, and we'll talk about crenellation later on when we talk about membranes. So you have lamins inside the nuclear envelope. Those lamins are A, B, and C. Lamin B can actually be physically attached to the nuclear membrane to hold it in place. These lamins can also be linked to the components of the cytoskeleton. So the lamins are inside the nucleus. Outside of the nucleus, you can have different components of the cytoskeleton. So outside of the nucleus, you have actin filaments, intermediate filaments, microtubules. Inside the nucleus, you have the lamins. There are structures called link complexes. So link complexes are numbers of different proteins, but their job is to link the lamins within the nucleus to different cytoskeletal components in the cytoplasm. So you can link the nucleus through these link complexes to the actin cytoskeleton, intermediate filament cytoskeleton, and microtubule cytoskeleton. So you're holding the nucleus in place, or moving it around if you wish. Okay, so lamins are intermediate filaments, and they're linked through proteins to potentially other intermediate filaments in the cytoplasm, or microtubules, or actin. Right? So intermediate filaments can support membranes. In this case, the lamins are supporting the nuclear membrane. But more importantly, intermediate filaments can support the plasma membrane. And that's really important in a lot of functions, particularly when we talk about epithelial cells, which we will in a couple of slides. So in this top right here, you see a picture of fluorescently labeled vimentin. So vimentin is an intermediate filament protein. So you have lamins in the nucleus, and in the rest of the cell, you have vimentin. Vimentin is found in the mesenchymal cell, so you know that this is a migrating cell that has vimentin. Vimentin can support the plasma membrane through an intermediate filament associated protein. In this case, it's anchored. So you have intermediate filament proteins right next to the plasma membrane, and they're supporting the plasma membrane by anchoring. If you remember, anchoring also supports the actin cytoskeletal membrane with spectrin. So anchoring can anchor the actin cytoskeleton, and anchoring can anchor the vimentin, or intermediate filament cytoskeleton. Okay, so, sorry, this microphone's moving around. So again, inside a cell, you're going to have intermediate filaments, microtubules, and microfilaments, or actin. These can interact with each other. There's lots of proteins that link these together, right? Often these proteins are also linked to membranes. The actin can be linked to a plasma membrane. Indeed, with stress fibers, the actin is linked to the plasma membrane with these proteins called integrins, if nothing else. Intermediate filaments can be linked to the plasma membrane. Right? So here you have intermediate filament bimentin. So this is probably a migratory cell. It has actin. This actin looks like stress fibers. That makes sense that this is a migrating cell. It has both bimentin and it has actin that looks like stress fibers. So it all makes sense. When you look at these and you think about this cell has to undergo mitosis, potentially you have to depolymerize all of these structures to undergo things like mitosis, because you have to remove all this so you can make a mitotic apparatus. So we know that you can depolymerize tubules, we know you can depolymerize actin. You can also depolymerize intermediate filaments. Intermediate filaments are dynamic. They do polymerize and depolymerize. They just don't do so very quickly. But you can see that they're in a dynamic state by sort of watching things as you add substrates. So in this experiment here, they've added biotin-labeled keratin. So you take keratin, which is intermediate filament substrate, and you label it with biotin so you can detect later. So biotin is just a biomarker. And you inject biotin-labeled keratin into the cell. If it's keratin, you know this is an epithelial cell. When you first inject it, you can see the biotin-labeled keratin is sort of present everywhere. If you wait a little while, you can see that that biotin-labeled keratin has now been incorporated into these intermediate filaments. So intermediate filaments do get polymerized and depolymerized. They are dynamic, just not as dynamic as actin and microtubules, and we don't necessarily understand how this is regulated. We do know one thing about the regulation, though, and that is the lamins that are present in the nucleus. During mitosis, you have to break apart the nuclear membrane. So to break apart the nuclear membrane, you have to get rid of those lamins. So during mitosis, one of the things that's involved are cyclin-dependent kinases. There's lots of different cyclin-dependent kinases. One of them is involved for phosphorylating a serine residue in lamin A. If you remember, inside the nucleus, there's lamin A, B, and C. A cyclin-dependent kinase phosphorylates a serine in lamin A, and when it does that, the lamin structure within the nucleus begins to fall apart during mitosis, for example. If you get rid of that serine residue, you don't get nuclear breakdown and mitosis can't occur. Right? So the regulation of lamins, at least, can be regulated by kinase activity and phosphatase activity. In this case, cyclin-dependent kinases can cause the disassembly of the lamins during mitosis. Yep.
Does that serine need to be phosphorylated on each individual tetramer to disassemble?
Does that serine, way too many details, probably, but too many details. How many serines do you really need to phosphorylate to get things to come apart? I think it just starts to destabilize the more you have as it goes. But too many details, unless you want that question. No. All right, so as I mentioned, one of the crucial roles for intermediate filaments is structural, and that's not seen anywhere better. That's almost English. You don't see that anywhere better than like in your skin. Your skin is made of epithelial cells. Epithelial cells have several jobs. One of them is to be a barrier. Right, so your epithelial cells in your skin, in your gut, wherever it is a barrier. It's preventing things from going through. So that barrier has to be strong. Cells are made of phospholipid bilayers as membranes. Membranes are not strong, right? You need proteins for that strength. If you just have cells, sort of even, membranes are easy to rip and tear. So the membranes in cells are strengthened through interaction with the cytoskeleton, particularly with intermediate filaments. And in case of epithelial cells, that intermediate filament is keratin. So keratin links epithelial cells together, and keratin links epithelial cells to subepithelial cells, right? So there's the deeper dermal layers and stuff. So when you pull on your skin, little bits of cells don't come off, it doesn't separate from the surrounding tissue because all of those cells have keratin within them, and that keratin is holding things in place. So in normal skin, in your epidermis, all of these epidermal cells are held together by keratin, and then they're linked to the dermis because keratin is kind of holding everything in place. The importance of that can be seen if you knock out keratin. So if you have a transgenic mouse where keratin is not functional, then you get blistering and easy separation of either the epithelial cells from themselves or from the dermal layer. So you need keratin. Without the keratin, your skin basically falls apart. Okay, 2 structures to memorize that we're going to continue with next lecture with cell adhesion, but these will be on this test. Okay, so there are different types of epithelial cell layers. Regardless of the epithelial cell layer, they're going to have keratin in them. That keratin is going to form structures that provide support. Okay, so keratin is like a rope, if you want to look at it that way. It can tie things together, so when you pull on one, you're pulling on a whole bunch of things. So in this little diagram, they've tried to show you such an organization of keratin with things. It doesn't really look like this. It doesn't really matter what it looks like. There's different representations you'll see in the next lecture, but this is good enough. So you have epithelial cells that have plasma membranes. The plasma membranes themselves are not strong. To form strength, you need proteins. So there are lots of proteins involved in cell-cell adhesion and cell ECM adhesion. We already talked about one of these proteins with cell ECM adhesion. Those proteins are called integrins. So integrins can link the cytoskeleton to the extracellular matrix. There's other proteins that can link cells together. Those are called cadherins, and we'll talk about cadherins later. So you have these proteins that are binding membranes to other things. So these are transmembrane proteins. So you can have transmembrane proteins between cells, and you can have transmembrane proteins between cells and extracellular matrix. So those transmembrane proteins provide strength, but they really provide strength if you link all of them together with intermediate filaments. In the case of these cells, these are epithelial cells, so the intermediate filament is keratin. So you have these keratin links linking all of these structures together. So if a keratin is linking cell-cell structures together, it's called a desmosome. So a desmosome is intermediate filaments holding cells together. So desmosome is a cell-cell adhesion. Desmosome is a cell-cell adhesion involving intermediate filaments. In this case, it's keratin, so it's an epithelial cell, but you have different types of cells. So desmosome, cell-cell adhesion involving intermediate filaments. A hemidesmosome is a cell ECM adhesion involving is a cell ECM adhesion involving intermediate filaments. Because it's an epithelial cell here, the intermediate filament is keratin. So you have desmosomes and hemidesmosomes that are holding cells in place, and they have these intermediate filament proteins binding everything together. This little diagram is sort of, I'm not sure why they drew it like this. If you can imagine the intermediate filament going from this desmosome to this desmosome to this desmosome to this desmosome, like if you can imagine sort of intermediate filaments linking all the desmosomes together, I think it's a better picture of how you provide strength. And if you then cross-link this desmosome to this hemidesmosome, etc., then you get a sense of how these proteins, if you have the like ropes, you've tied everything together, then when you pull on one cell, you're pulling on everything else, and that structure holds together. Okay, so intermediate filaments are involved in cell integrity. They're involved in cell-cell adhesion structures called desmosomes, and cell ECM adhesion structures called hemidesmosomes. And we'll learn much more about those next lecture when we talk about cell adhesion, but these structures and intermediate filaments will be on the test on Friday. And this is where we're ending. All right, so good luck Friday. I do have office hours today and on Thursday, tomorrow. The carrot is binding the desmogens to lots of other things. The desmogens are binding cells together, and the carrot is binding to lots of other
So basically, like you just explained. So basically,
I was cutting. I cut the teeth. That's correct.
between the
So the way they've done it,
it's kind of weird.
yeah. In the next lecture, there's other diagrams that here they have.
Jesus, you guys are crazy.